This video is beginning the first part of chapter 9 on microbial growth and how microbes grow. We have pictures here of the biofilm to start out the chapter, and that's going to be covered in our very first section. So we'll talk about biofilms, what they're made out of, how they protect, how some of them are good for us and others are not so great for us, but they can grow on pretty much any surface. It could be solid surface, it could be floating out in the water, but biofilms are out there and they are accumulations of different types of microorganisms that work together. So let's start with how they grow. Our case study is Jenny. She's 24 and she is pregnant in her second trimester. She's got a fever. She's running 102, fatigue, muscle aches, kind of flu-like achy symptoms. Overall, she's pretty healthy. She's exercising regularly. She's got a nutritious diet, organic foods, and raw milk that she purchases locally at a farmer's market. All of her shots are up to date, and we're worried about her symptoms. So we're going to take a blood sample and send it off and see what's going on with Jenny. Okay, so how do they grow? This is the whole point of this section, is talking about bacterial growth. So how do they grow? The most popular kind of growth in bacteria is going to be binary fission. This is the most popular kind, and it's similar to in eukaryotic cells when we talk about mitosis, binary fission for bacterial cells. So the DNA is going to replicate at a place called the point of origin, and that DNA is going to copy itself, and eventually the DNA is going to split. After the DNA splits and it goes to its separate sides, we need to form the wall. The wall is going to be made out of the FTSZ proteins, and they are going to come together, and eventually it's going to form a little wall, a little septum, and then eventually we're going to fill that in with a real cell membrane and a real cell wall and divide it out. And now we've got the two brand new daughter cells that are absolutely identical to the original cell. This is mitosis for bacteria. It is binary fission. It is copying that circular DNA and using FTSZ proteins to create a wall that eventually splits the cell in half. So when we're undergoing binary fission, we go from one cell and we produce two cells. How long does that process take? That is generation time. The amount of time that it takes for a population to double from 10 to 20, from 1 to 2, from 2 to 4. So if we have different bacteria have different types, different times. We've got E. coli. E. coli grows if it's in a medium that it's happy with, that hits all of its needs for metabolism requirements. It can double itself in 20 minutes. 20 minutes, people. If we were talking about TB, TB is a lot slower. It's not 20 minutes, it's 20 hours. And then even slower than that is Mycobacterium leprae, which is leprosy or Hansen's disease. That can take up to 14 days before it doubles in population growth. So here we start off with our inoculum and we only got literally one cell. And that one cell is going to go through its first generation time and go from 1 to 2. And now let's do another generation time. We're in the second generation. Those 2 cells are now going to become 4 cells. And by the third generation, 4 cells are now going to double and become 8 cells. So if this was E. coli, this would be 20 minutes. This would be 20 minutes. This would be 20 minutes. And in an hour, we've gone from 1 cell, 20 minutes, 40 minutes, 60 minutes. In an hour, we've gone from 1 cell to 8 cells there. So you might have some sort of basic math problem like that on the test. I'll cover it in test review if we do, but generation time is basically doubling time. It's another name for doubling time, where that population literally doubles. How long does it take? It depends on the organism. Some are really fast and some are slow. When they are doing this, we can track this mathematically on a growth curve. So there are different places on here that I want y'all to know. I want y'all to know the different phases of the growth curve. So the very first phase is called the lag phase, and that's where you've just put the bacteria and inoculated it into your nutrient media. You just did your broth to broth transfer and you put it in the brand new media, and now you're in the lag phase. You're waiting. So what happens? That bacteria is sensing what's all around it. What kind of media am I in? What food do I have to choose from? What kind of different sugars can I metabolize? What's out there for me? Let me figure it out, get my stuff in order, and then I'm going to grow. So the next phase, after I've gotten my stuff in order, is the logarithmic growth phase. It's exponential growth. So you have this J curve going on. That's exactly what we're seeing right now with COVID. We have this J curve of number of infections, exponential growth. 1 to 2, 2 to 4, 4 to 8, 8 to 16, and on and on and on. That is logarithmic exponential growth. So this is the log phase where the bacteria is just going gangbusters. It is loving what it's in this closed environment. It's totally happy, and it's got logarithmic growth, exponential growth. But eventually, we're going to hit a stationary or a plateau phase. On the plateau phase, again, this is a closed environment, so we are eventually going to slow down on all those sugars that we had available because we've used up everything that's readily available, and we're going to start to slow. So our rate of cell division is going to slow down. So the rate of growth is going to equal the rate of death. And then we are going to move past that stationary plateau phase to the death phase, which aptly named the rates of cell death is going to be greater than the rates of cell growth. So the species is going to decline. Sometimes in this death phase, uh, the cells realize that the conditions are bad, and so they're going to slow down their metabolism. They will shut stuff down and kind of go into hibernation mode, and sometimes those guys are called persisters. So they can hang out in this hibernation state until conditions get better. So these persisters lead to chronic infections. You start to feel better, the bacteria you think is gone, and then later, these persisters that were lurking come back out, and now you've got another flare up of this infection going on. So persisters are hanging out in this death phase, not dying. And it mentioned tuberculosis is known for these, is known for persisters. So in this bacterial growth curve, understand that this is a closed environment. We're using one broth tube and it's sealed up, and whatever's happening in there is happening in there. Here's our logarithmic growth. We can see it on the arithmetic scale here in A, where it's got this J-shaped curve, or we can do it in the semi-logarithmic scale. Why isn't it working? There we go. And we can plot it, you know, x-axis, y-axis, x-axis, y-axis, and it looks more linear that way. But this is exponential growth in the log phase of the growth curve. On our bacterial growth, we said previously in this slide that we're in a closed system. Whatever nutrient media you have there is it. There's something called a chemostat, and with a chemostat, you are constantly making sure that that culture, that media that you're putting your bacteria into is new and fresh. So these are pretty cool. They are going to add in fresh media at the same amount that they take out the used up waste filled media. So there's always new stuff in there. Air is going to be brought in and mixed. So if it's a bacteria that needs to use aerobic metabolism, they're always getting a fresh supply of air. The oxygen's not running out. So this is a chemostat, and if you need to grow a certain species for an extended length of time, the chemostat is the way to go so you don't run out of your stuff. It never goes down into the death curve because you've always got fresh stuff going in there. Ways that you can measure and count these little guys, these bacteria. We have the first group are going to be called direct cell counts. So with this one, we've got the Petroff-Hauser chamber. And so you can see it is this plastic slide, and then you would put your cells on there. You're gonna put your culture on there, and you can, when you zoom in on it, when you increase your magnification, you will be able to see this picture over here. And based on the lines on this slide, you can count out, you can physically count out the number of cells. So this is a method of direct cell counts using this Petroff-Hauser chamber to count out how many cells that you are seeing in here. There's pros and cons of the direct cell count. With this guy, you're seeing them, but you don't know if those cells are alive or dead. With the fluorescent staining that we can do now, we can use the fluorescent stain to distinguish. The green ones are going to be all the cells that are still alive. The red ones are going to be fluorescent stain that are on cells that have their membrane that are damaged. So for every red cell, that red stain is going to take over, and the red ones are going to displace the green stain. So every time we see this red one, we know that that's a dead cell. And that helps to differentiate, because if you're just counting these all up, how many of them are alive? How many are dead? Am I looking at everything that's dead? Is this a culture of dead stuff? And if I do have a culture of dead stuff, if I'm treating it with some kind of drug and trying to see if it works, if everything's already dead, it's not helpful to me. So I need to know on this stuff what's alive and what's dead. Who's going to be able to undergo metabolism and show me things, and how many are dead. So this fluorescent method of staining with the green and the red dyes is really helpful, more so than just being able to count the cells. Now we can see who's alive and who's not. Another type of direct counter is the Coulter counter. So we had mentioned before on our chapter talking about cells and cell parts, that the cell membrane is going to keep some things in and some things out, and the cell is going to keep an electrochemical gradient, a charged difference between the outside of the cell and the inside of the cell. So that electrochemical gradient, we can be able to measure this with the Coulter counter. So it's got an electrode over here, and it's got an electrode over here, and we got a positive and a negative side. So just like a battery, and it's going to run a current between those two probes. And then in between, you're going to have this guy right here and this guy right here, these tube. And as the bacteria pass between the tubes, it can sense that difference in electrical current, and it counts it as a cell. So this is the Coulter counter, and every time the cells go through this teeny little glass tube, it can measure that difference in electrical charge, which is kind of neat, and that helps us figure out how many cells we've got going on in this type of direct count. This is the Coulter counter. Another way that you can do a direct count is a plate count, and you can do that with a serial dilution, because a lot of times stuff, if we just took it straight out of the pond, say for example, usually we do this with some pond water. If we took it straight out of the pond and we grew it, it would look like this. There would be so many bacteria growing in that straight, direct, concentrated solution out of the pond that we can't count those. There's too many to count. So what we do is a serial dilution. So here we are going to have a tube, and in that tube we have 9 mils of water that we're going to dilute with another 1 mil of stock. So together 9 mils and the 1 mils is going to bring 10 mils total. And then you're going to put those guys, your 9 mils of water, your 1 mil of your stock solution, and you put it on the stirrer that we've got in the lab, and it would spin it up, stir it all together. Now you've got 10 mils of a solution, and that dilution is at a 1:10 ratio. So you took 1 mil out and added it with 9 mils for a total of 10 mils. So it's a 1:10 ratio. And then if you plated that, again, it's so concentrated, it's going to be too much to count. It's just going to overgrow your plate, and you can't count individual colonies. So let's dilute it again. We're going to do the exact same thing. We got 9 mils of water over here. We're gonna add in 1 mil of our concentrated from this guy. So not from our original stock. We are now pulling from the 1 to 10 ratio. And we'll pull 1 mil out, add it to the 9 mils over here for a total of 10 mils. And so now we've diluted it another 10 times. So now it's again another 1 to 10 ratio times the original 1 to 10 ratio. So we are at a 1 to 100 ratio at this point. So it was 10 over here, and then 10 times 10 again, it's going to be 100. And then we could take a sample from that, grow that, and it's still too much. I still can't make out individual colonies on the serial dilution at 1 to 100. So let's keep going. Repeat the process. Pull out 1 mil, add it to 9 mils of water. We're at 1 to 1,000. Pull out a sample of that and grow those. And if you grow those, we've got 389 CFUs, 389 colony forming units that we can count. That's too many. Anything above 300 is just too many. It's overcrowded and you can't count individual colonies. So we're going to go again. We'll go one more time. So we'll pull 1 mil, put it in with another 9 mils. So we're doing another 1 to 10 dilution. For a serial dilution, last time we were at 1,000. Now we are at 10,000 because it was 10 times 1,000. So 1 times 10,000 ratio. We're going to pull 0.1 mils out, grow it, and now we got 50 colonies. That's a magic number. Our magic number has to be somewhere between 30 CFUs and 300 CFUs. So at 50 colonies, we can count out, and that's a great serial dilution. Let's see what happens if we dilute it one more time. So if we pulled 1 mil out of here, added it to the 9 mils of water, we're at 1 to 100,000. We're only getting 2 colonies, and that's not enough. That's too sterile. So our best ratio with this stuff was when we found the 50 colonies. I was at 1 to 10,000, and we can actually count those. So this is how a serial dilution works. We do these virtually in Labster instead of doing them face to face, but it's the same process. You're going to take it out, you're going to dilute it with water, then you're going to pull a little bit of that, put that in, dilute it with water, pull a little bit from that vial into the next vial with some water, on and on and on. So we have a 1 to 10 ratio every time we diluted this pond water in our serial dilution. And then we put it in a plate and we grew that plate. How many colony forming units did we see? Too many, too many. 389, still too many. 50, that's a good number, anywhere from 30 to 300. And then 2, that's not enough. It's too small. But this is a serial dilution, and we are putting it onto a plate. We were talking about the numbers and how do we calculate out those CFUs. So if we were using the 1 to 100 plate and we found 148 colonies growing at that level, then we times it by 100, and that gives us 14,800, 14,800,000 CFUs in 100 mils. We were pretending, we always look at CFUs per 100 milliliters. So that's why we add this extra 100 over here. So if we have 14,800,000 colony forming units in 100 mils, that is well above the 4,000 max limit where you would want to be hanging around in this water, boating and fishing. That's way, way above. You have way too many E. coli CFUs growing in that water. So definitely don't drink it. Don't go swimming in it. And don't even boat or fish in it. Don't do it. At least not with this sample, because that sample was gross. So that's how we count it. That's how we measure it in a serial dilution. When we pour these things, there's 2 ways that you can do it. You can do a pour plate method, or you can do a spread plate method. Both of them involve a plate, your agarose plate, and you're going to pour it on there. But this one, you pour it on, just like it says right here, pour on the sterile plate, and then you swirl it. So you're going to grab the plate and you just kind of tilt it. You'll tilt it, and watch it pour all around, and you'll swirl it all around and try your best to coat the plate that way. This is a pour plate method. You're not touching it. You just hold up the plate and you tilt it and make sure that it gets all over top of it. And then you'll count the CFUs that grow from that about 48 hours after you pour your plate. The spread plate method, pour it onto the plate. And instead of just holding your Petri dish and tilting it and hoping that you get all the liquid solution all over the plate so it makes a nice even spread, here you're going to use the spread plate method actually uses a spreader bar. So you would get this spreader bar right here, a sterile one. It comes out of the package, and you have your plate, and here's your spreader bar, and you make sure that you spread that liquid all around the agarose with the bar. So there's two methods: pour, no bar. Spread plate uses a spreader bar to get the liquid all around on your agarose plate. Another method of calculation is called the most probable number MPN. So we said that different bacteria have different types of metabolism. They can handle all sorts of different types of energy sources, different sugars, other types of substances, and sometimes they may be able to handle one sugar and not a different type of sugar. So in this one, we are setting it up to look for colony forming units, and we are pulling them and putting them into a lactose solution. It's got this lactose reagent in here, and if this colony forming unit, if like E. coli, for example, if it can metabolize lactose, that red dye, and I can't remember what it's called, it has a name, but the red will change from red to yellow. And so if it metabolizes that sugar, that reagent will change color because of the acidity that happens when it metabolizes, and it changes from red to yellow. So we definitely got 5 positive tubes in this instance because there were so many bacteria in this 10 mils. When we inoculated each one of these with 10, 10, 10, 10, 10, all of them turned positive. So let's take less out. If we use fewer bacteria out of our container and we only pull 1 mil out of our stock condition, and we add 1, 1, 1, 1, 1, what happens? Are all of them going to change? Are none of them going to change? And in this case, we got 2 tubes out of the 5 that changed. So let's see if we dilute it again, what happens for our most probable number. So in this one, we have 0.1 mils that we took out. So we're going down by levels of 10. We started with 10, then we used 1 mil, and then we used 0.1 mils. And on this one, 0.1, 0.1, 0.1, 0.1, 0.1, and none of them changed color. So we didn't get any kind of reaction when we took that teeny tiny little bit of substance of bacteria out of our stock. So let's count up the numbers. At 10, 1, and 0.1, we got 5, 2, and 0. What do I do with those numbers? What is that going to tell me? So we're going to take those numbers. We're going to take those numbers and we're going to plug them into this most probable number table that they give you at the back of the textbook. And so I did it for you and highlighted it. And here's where we're at. Here's the 5. Here is the 2. And then here is the 0 at our 10, 1, and 0.1 levels of concentration. And what it tells us is out of every 100 mils, there are 49 estimated, most probable 49 bacteria found in the 100 mils. So that's our number given this test with our most probable number at 10, 1, and 0.1, that at about every 100 mils, we got 49 bacterial cells. 49 bacteria cells using this type of test. Another one that we can use to test are indirect. So not actually seeing the physical cells and counting them up, or the colonies and counting them up. Now we're going to see what the colonies are doing to their environment. So we're not seeing them, but we're seeing evidence of them indirectly. So one of those methods is turbidity. Turbidity is cloudiness. That's something that you can measure with a machine called a spectrophotometer. A spectrophotometer is something that if you were in Majors Bio 1, we use this. We use this a lot in Majors Bio 1. If you did the photosynthesis lab that I mentioned in the previous chapter, if you were able to do that in your Majors Bio lab, I believe we also do it for non-majors, possibly at the Cartersville campus. What we did was we were looking for those pigments in photosynthesis. We were seeing where chlorophyll A and chlorophyll B and the carotenoids, what wavelength were they absorbing? And you could do that with a spec machine. So the spec machine is going to measure transmittance, how much light goes through the material. If we were looking through water, it's going to transmit better than if we were looking through a can of Coke. That would not transmit better. So here we've got very clear solution. We've got brand new media here. Nothing's in it and it's clear, versus over here, this media is chock full of bacteria. And when it's growing in the bacteria and it's really happy in that media, it gets cloudy. It's not clear anymore, it's cloudy. So we can measure that turbidity, how much light is not getting through. It's not getting through with transmittance. It's being absorbed by that cloudiness, and we can measure that turbidity with a spec machine, with a spectrophotometer. Another way that we can try and figure out how many we have growing, we can use alternative methods of cell division. That's another way that we could maybe possibly diagnose what's going on. We said most popular methods of cell division were binary fission, but there's also other methods of cell division for bacteria. We've got the cyanobacteria, and they're going to use fragmentation. So basically a piece of it breaks off and it copies itself asexual reproduction, just like binary fission, but instead of copying itself and splitting in half and perfect split in half with fission, where it just divides in half, even Stevens, fragmentation is where a chunk of that parent filament of the cyanobacteria breaks away, and it will grow up and develop on its own. Then we've got, what is this one? We've got Gemata, and it is undergoing budding. So Gemata does budding, yeast also does budding, and you're going to have a parent organism, and that parent is going to bud off a little baby version of itself. So those are different types of cell growth that we can use in addition to if they don't use binary fission. Biofilms are our last topic in this section for the chapter. And biofilms are important. You see these guys everywhere. They're their own little ecosystems. So they are going to be composed of different species that work together. They work together in different forms of metabolism. So say that you've got some aerobic types of bacteria, and they are going to need oxygen, so they're going to pull out all the oxygen in the local tissues and in that area, and then they are going to hog up all the oxygen. So then if there's another bacteria in there that likes an anaerobic environment, it's a good thing that the aerobic bacteria were there, because they pulled out all the oxygen, and now you've made an anaerobic bacteria really happy. So they can buddy up together. If you had the aerobic ones on the outside and the anaerobic ones on the inside of the biofilm, it works. If one bacteria metabolizes the byproduct of another bacteria's metabolism, it's symbiotic. So these things are different types of species that work together, and they form a biofilm. That biofilm is covered with, does it tell us in here? Yes, the EPS, that's what I'm looking for. So it's covered with this extracellular polymeric substance, EPS. It's basically a polysaccharide sugary coating around the biofilm. So the biofilm's EPS could be half of the material or as much as 90% of that EPS. And so it's going to kind of act like the housing of the biofilm, and they'll form little chambers in there where they can get fluid running from different groups of bacteria. And so the nutrients can get through and the wastes can get out through channels in this EPS stuff. So we have steps of biofilm formation, and we've got the first steps are where they start to attach. So we've got free floating plankton going on, and they are going to need to attach then they become irreversibly attached. So they permanently attach. Dog party. Sorry. They permanently attach. Then we're going to undergo cell division. They grow, 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 grow. They're going to form that sugary coating on the outside, the EPS. They're going to form their water channels that they'll be able to get the nutrients and the waste through. And then we've got our secondary attachment where colonizers are going to go out and the cycle starts. Again, those are the steps for biofilm formation. So again, we have these everywhere. Sometimes in a biofilm, we're going to have quorum sensing going on. And this could be a good thing or a bad thing. We have biofilms that are in our gut and our respiratory passages that help fend off pathogens. So those are good biofilms. But there are also biofilms in places that we don't want them. Like maybe we get a biofilm growing on a catheter, or like my brother-in-law had chemotherapy and he had a port put in. And so he had an infection, a biofilm growing on that port. And so he had to have that removed. So sometimes biofilms are not good and we need to be able to tackle those. And it's hard to tackle those because sometimes the population will get where it has the numbers that it wants and it'll sense that with quorum sensing, and it may start to release and be pathogenic when it wasn't pathogenic before. If we're trying to treat this pathogenic biofilm, it's hard to treat them. That candy coating protects them. The cells that are on the inside of that sugary coating, they've slowed down their metabolism and maybe they're not even bringing in the nutrients right now. Maybe they're kind of dormant. And so even if you covered them with an antibiotic, something that could possibly kill them if they brought in this medicine, if they don't bring in the medicine, that's not going to work on the biofilm. So they the biofilm, that candy coating, is going to slow the medicine from getting to them. Even if it does get to them, if their metabolism is shut down on the inside of the candy shell, they're not going to bring in that medicine. Sometimes a biofilm will cause the different bacteria in there to increase the number of pumps that they have in their membranes, and that physically ejects stuff that could get pumped into the cell. They just pump it right back out and it doesn't bother them. So we'll talk more about that kind of stuff later in the semester. But biofilms, once they get established, are tricky and hard to overcome when we're talking about in the medical films, in the medical field, on a medical device. They're hard to get rid of. These are the different types of quorum sensing molecules. I'm not going to ask you about that, but I do want you to know what quorum sensing is, and that's where there's a lot of them. They finally get to that magic number, and now it's go time to express whatever genes that we might not have expressed before when there was just 2 of us. Once we finally got 100 of us, oh, well, now I'm going to express that toxic chemical out there and make my host really, really sick. Or maybe I'm the bacteria that lives inside the little squid that lives outside of Hawaii, and now it's time to glow. It just depends. But quorum sensing is when they sense others, and once they finally get to the magic number for them, they do whatever they need to do. So that covers the first part of chapter 9, and I'm gonna take my dog out so she